a very powerful laser. So it's great to be back in Singapore. I love coming to Singapore, mainly for the food, but also because the people are very kind and friendly, and they believe in science, which is good. So Stefan Hell, I met him. We were together at a meeting in Hong Kong recently, and I think he's a really, really good example of how if you want to win a Nobel Prize, the best way of doing it is to do something which everybody believes is theoretically impossible, right? I mean, there are a number of other things. When I was a student, DNA sequencing was theoretically impossible. Understanding the structure of the ribosome was theoretically impossible because ribosomes would, A, never crystallize, and B, if you crystallized them, you couldn't solve the structure. It was beyond the power of computing. So, and I want to try to explain to you how sort of, that's sort of why I won a Nobel Prize too, I guess. I'm not sure, because you'll see, you know, people talk about brilliant minds, but actually we're just ordinary people. I mean, my wife says, you know, if I can win a Nobel Prize, anybody can. So the story of my life, I grew up in Oxford and spent most of my young days until I was sort of 12 or 13 years old learning Latin and Greek, at which I got progressively worse and worse. And fortunately, I had a very good science teacher. We only had one science lesson and probably about 20 or 30 Latin and Greek lessons every week. It was terrible. And this man, he's no longer with us, Gert, told me that I was a good biologist. I wasn't particularly interested in biology. I was just good at it. It was sort of a strange thing. I did very well in an exam. And that I knew from then on. And I was helped too by the fact that my best friend was a wonderful physicist, and he showed me very clearly that I was no physicist and would never, I would have loved to have been a physicist, but I wasn't. So that, you know, there you go. And then I sort of passed the exams and got into Cambridge. And I think probably the most important lesson that I was taught there was not to believe anything that people told you, or whether they told you in person or whether they told you in print. Find out for yourself. Make sure that your feet are planted securely on the ground before you take the next step. And, you know, I often say to people I was born with a sort of scientific silver spoon in my mouth because I was an undergraduate from 61 to 64 and a graduate student from 64 to 68. And of course, you know, the great pioneers, Fred Sanger, Francis Crick, Sidney Brenner, Max Perutz, they all came, they taught in the University of Cambridge. I knew them. I would sometimes sit next to Francis Crick at dinner, and you know, this was an extraordinary privilege. And you know, what that taught one was that these people were perfectly ordinary. I mean, they were like you and me. And the only thing is they'd done something absolutely amazing and turned over stones and revolutionized our ways of looking at the world. And I think the one thing that I thought I detected that they had in common was they liked things to be simple. And then there was a certain humility. I mean, you know, there was, like Sidney Brenner is an incredibly clever person, but Fred Sanger, if you met him, was won, by the way, 2 Nobel Prizes. You would have taken him as the janitor or the gardener or something like that. I mean, it was very, although when he got his second prize, apparently, I wasn't there, but there was a party, and he said, people think that I'm very modest, but actually I think I'm bloody good. Anyway, so that was good. And then the question is, you know, so I became a graduate student, and my supervisor said, you know, Tim, I said, what should I work on? He said, well, why don't you go to the library and find a project? So I went to the library and found a project, and the project didn't work because I didn't read the materials and method. I used the wrong kind of rat, and it didn't work. So after 6 months, I had absolutely nothing to show. But I had sort of learned what was in all the drawers and how to pipette and things like that. So that was useful. And then really my entire career was based on talks my first scientific meeting by these two people. And what did I learn? The whole meeting was about hemoglobin, structure, function, and synthesis. And what Borsuk had discovered, with a man called Jacques Creux, a Frenchman, was that iron controlled globin synthesis. And at the bottom here it says, it might have been expected that heme synthesis would be accelerated by iron, but hardly globin synthesis. It's a very surprising result. So there was coordination. If there wasn't any heme, the globin synthesis stopped. Okay? And the other talk that I had was from Vernon Ingram, and he was studying that problem. And what he said was the amazing thing that ribosomes went up to the point where they synthesized the fold that heme was going to fit into. And if the heme wasn't there, they sort of went along the ribosomes and then looking for the heme, where's the heme? Where's the heme? And they stopped waiting for the heme to come in. Pretty amazing. So I went back to the lab, and Lou Reichard, who's a very fine biologist and a lot cleverer than me, pointed out that when I explained the data that Ingram had showed, that actually Ingram, a great scientist, had totally misinterpreted his results and got it backwards. And at that point, I can't quite reconstruct what happened now, but basically what happened was that we decided to find out for ourselves whether the ribosomes formed cues. And to cut a long story short, and the long story also involves this guy on the right here, Tony Hunter, also a very fine scientist, still working away at the Salk Institute, by the way. And Tony and I sort of formed a consortium to do our PhDs together, and we looked at this point, and we found that the ribosomes didn't form a queue, actually. Although if we wanted to, we could make them form a queue, and our methodology could detect the cues. So that was all cool. And during that time, somebody just up the road, unbeknownst to us at the time, found that if you took these reticulocytes, which are called so because they have this sort of lacy network which is revealed by certain basic dyes, and you spun them down in a test tube and washed them and then simply added water, they would burst open and you had instant access to the cytoplasm inside, and they would continue to synthesize globin, and that synthesis was dependent on heme. So now we had access to the heme control system. And I ran into this guy at my second scientific meetings. The moral here is that it's jolly good to go to scientific meetings because occasionally you meet people who will change your life. He's called Irving London, and it turned out that he was very interested in the same thing, although actually his sort of day job was being head of the Department of Medicine at the Albert Einstein College of Medicine in New York. And here is the kind of thing where I ended up as a postdoc with him. And you see, this is now measuring radioactivity in proteins, and you can see that if you add the heme, this is pretty much the same rate as the cells would have been making protein if you hadn't burst them open. It's a remarkable fact, that, and I still don't quite understand how it works, but let's not go there. But if you leave out the heme, protein synthesis starts off all right and then sort of curls over and dies to a low rate, typically a 10th or a 20th of the initial rate. It never quite stops, it just slows down a lot. And if you add back the heme, I can't remember when the heme was added back here, then it recovers, so the control is reversible. So how the hell does it work? Well, we could repeat, this was an incredibly repeatable experiment, and one of the advantages of the system, you could freeze this reticulocyte lysate in liquid nitrogen, so you could do an experiment any time of the day, any time of the night, any day of the week. And if you had a good idea, you could do about 3 experiments a day, given it was bloody exhausting, but you could. So it was very quick and easy. I really admire you people who sort of grow cells and have to wait for days before you get a result. That's awful, actually. I simply couldn't do it. I would have forgotten what the experiment was by the time it... Anyway, so I sort of worked away, and then Irving went away to found a new medical school, and I was left alone with a technician, and I started doing sort of naughty experiments on the side after the technician went home, after dinner experiments, Saturday morning, weekend experiments. And I won't go into why I did it, but Ellie Ehrenfeld and I discovered that tiny amounts of double-stranded RNA inhibit globin synthesis. We were trying to translate poliovirus, and that didn't work. So here's double-stranded RNA. It looks kind of like double-stranded DNA, but you can see the structure is quite different. And the amazing thing was that one molecule of poliovirus replicative form double-stranded RNA could inhibit all the ribosomes in a HeLa cell. So there's no way, this is 10 million ribosomes being stuck because of one molecule. And there isn't room so it had to be, first clue, it had to be catalytic. Somehow double-stranded RNA activated an inhibitor. And we were pretty dumb, actually. I didn't really articulate that clearly enough at the time. So my postdoc, although I discovered one or two other things as well that did the same thing, but we didn't get to the bottom of it, and so had to go back to Cambridge, where I still had a fellowship, and took a five-fold cut in salary. I'd been earning $11,500 a year as a postdoc in the medical school, and went back, I think, to £800 a year in Cambridge, and it wasn't a lot. But I did get free dinners because I was a college fellow and teaching a little bit. And Jim Watson says that you should always try to work with people who are cleverer than you are, and I think that's excellent advice. So look for clever people. And Richard, alas, is no longer with us, but he was a wonderful, wonderful collaborator, and we made a great team together. And we solved the problem, as I shall describe. So an inhibitor forms and it inhibits initiation. So how does initiation work? Well, at the time, Richard and Tony had just discovered that it started with a special initiator tRNA carrying methionine. So we started using radioactive methionine as the label, and as a result, made a great discovery, which was, I sort of apologize for this slide. I used to run hundreds of these every year. These are sucrose density gradients. And this is the top of the gradient. This huge peak here is hemoglobin. And then these are the small ribosome subunit, the large ribosomal subunit, single ribosomes, 2 ribosomes attached to message, 3 ribosomes, and the rest have run off the bottom. And what a graduate student found was that when he left the heme out, there was this big, unexpected, and mysterious peak of methionine over the small ribosomal subunit. And just before protein synthesis curled off and died, that peak disappeared. So pretty obvious, that peak has got something to do with how ribosomes find the start of messenger RNA. Now, this is very controversial because everybody knew that the way that protein synthesis works is you have a messenger RNA, and the messenger RNA tells you which tRNA to put in, right? Next comes an alanine. Next. Here, there's no messenger RNA here. Yet this bloody great big peak of methionine. It was very quick and easy to show that it was an initiator tRNA. So we had the bizarre thing is that actually the small ribosomal subunit binds to tRNA not under the instruction of messenger RNA. Actually, it uses the anticodon of the tRNA to look for the start of the messenger RNA. And when we submitted this paper to the Journal of Molecular Biology, which was kind of the Cell of its day, people said we just didn't understand the first thing about protein synthesis and we got it all wrong. Always been my experience that if you really find out something important, the referees will give you a very hard time. So you better be bloody sure that you're right, because otherwise they will kill you. So this is it. I mean, this is now molecular biology of the cell, Keith Roberts, excellent, and Nigel Orme's thing, right? The small subunits gets loaded with initiator tRNA. The initiator binds to the messenger and scans along until it finds the first AUG, and then it stops. And then the large subunit can come in, and you can get conventional protein synthesis going. So this was really quite a paradigm shift, but we still didn't know how heme, or double-stranded RNA, or yet another thing, actually worked. And we got very confused. An Israeli scientist reported that cyclic AMP, here high levels of cyclic AMP overcame the inhibition, and we tried other things. I just realized that caffeine in your coffee was a good analog of adenine, and which I actually mostly use 2-aminopurine. And this is the kind of thing, so look how confusing this is. So here we have double-stranded RNA added at 0 time, shuts off protein synthesis, just like leaving out the heme. Add in the anti-inhibitor, 2-aminopurine. There's a lag, and then protein synthesis takes off. Pretty reversibly, actually. I mean, that's really good. But if we use heme, we add the 2-aminopurine, and protein synthesis is instantly reversed. Whereas if we add back heme, protein synthesis is only reversed very, very slowly. So what the hell is going on here? And we were just utterly baffled by all of this. And then, luckily for us, the lab burned down. And why I say luckily, for several reasons, actually. I think the most important thing was that all this confusing data was burnt up. And that was really, really good. We sort of, we just made a fresh start. But more to the point was that, I don't know, the gods were looking at us very kindly, and we were moved to a teaching lab up in the hospital, which was right opposite the famous laboratory of molecular biology. And Max Perutz took pity on us and said that we could use his stores, which were much better than the biochemistry stores. I mean, fantastic stores, anything in them. And perhaps the greatest gift of all, that we could have lunch in the MRC canteen, where, of course, there were all these amazing heroes of, and Francis Crick would come and sit down at lunch and explain about nucleosomes. And he was very concerned that everybody really sort of was on board. Sidney never came to lunch because he and Mrs. Perutz didn't get along. So there was that kind of stuff. And so it was a really, really good time. And I collaborated a lot with people from John Gurdon's lab and chatted to other people. So there were all these people in Cambridge. I mean, we didn't think of ourselves as geniuses or anything like that, but we did have interesting projects which we explained to one another and, made suggestions, made criticisms. And, in a way, science is really easy. You just sort of have an interesting problem, and you push it around, and you try and solve it. And you put one foot in front of the other, and somebody invents a new technique, and that allows you to make some progress or something. I mean, it was a wonderful experience. And as a result, very quickly, actually, we found that the inhibitor which shut off protein synthesis was a family of protein kinases that phosphorylated the very factor that binds the initiator tRNA to the 40S subunit. So that was sort of it. I mean, and now we know there are 4 of these things, and I won't go into all of that. That's another story. But you've got a problem. You've solved your problem. So now what do? Well, I remembered Borsuk's talk, and that the other half of his talk was about how when sea urchin eggs are fertilized, they turn on protein synthesis. So I'd been working on how you turned off protein synthesis. How about looking at the reverse? Problem, however, is that there are no sea urchins in Cambridge, or anywhere near Cambridge for that matter. So luckily for me, we organized an EMBO meeting on translational control. And remembering Borsuk's talk, I invited Tom Humphreys to come, who worked on sea urchin eggs, and was the only person I could find in the literature who still worked on this problem and hadn't made much progress. And it turned out that he was a keen cyclist, and so was I at the time, and he wanted to get a bike and said where could he rent one, and I lent him my bicycle. And we became friends because I lent him my bicycle. And he said, Tim, why don't you come and help me teach the course in Woods Hole next summer, and we can do some experiments on sea urchin eggs. So I leapt at the chance and went and taught that summer in Woods Hole. Didn't make much progress because although I had assumed that an American lab would have great facilities, They didn't have minus 80 freezers in the embryology course at the time. They didn't have liquid. The neurobiologists down in the basement did, but they were pretty standoffish and they weren't very cooperative, so that didn't work. And so I vowed I would never go back there because it wasn't good. But then the chance came a couple of years later, and I did go back, this time well prepared, and I took a suitcase. I mean, in those days, you could put, I would take my reticulocyte lysate with me in the whole, you know, the overhead baggage, you know, big styrofoam box full of carbon dioxide frozen. You can't do that anymore. It's really a shame. And we used to use tobacco mosaic virus as our test message. And I also had a little tube of that in my pocket. It was very robust and didn't tell the immigration. I was very naughty. And it's a beautiful place to go. And what is especially beautiful about it is that all the biologists going to Gordon conferences stopped by there on the way, because it's a very nice place to spend the weekend. And they would give seminars. And so absolutely, my entire biological education came from hearing talks. And you could hear talks morning, noon, and night. And wonderful, wonderful things over the years. And they had sea urchin eggs. And if you give a sea urchin egg a 12-volt alternating current electrical shock, it will shed its eggs. I don't recommend this. It's much better to cut them open with scissors and just scoop out the eggs, if you like sea urchin eggs, but which you can get in Japan. And we now export most of our sea urchins to Japan, because that's where they appreciate them. We don't. If you add sperm to those eggs, they start dividing. And they divide amazingly synchronously. You don't have to do anything except make sure that they're well stirred up to begin with. Just make sure that every egg gets a sperm. And that's easy to do. You just have to titrate the sperm and titrate the eggs and get the ratio right, and they'll all develop. That one isn't developing because it's an oocyte, and oocytes can't be fertilized if they're sea urchin oocytes. So that was good. And we did experiments, and we learned how to analyze the proteins that were made, but we didn't make much progress in figuring out. And the trouble was that these sea urchin eggs are only available in July, basically, and then the season is over, and there are no more eggs. So I would build huge castles in the air from August through June, and then go and test the hypothesis that I'd elaborated during. It was always wrong. You know, the very first experiment proved you were wrong, and then you didn't have enough time to really think about what was. So it wasn't super successful. But luckily for me, in the second summer there, I helped Eric and Joan with experiments on clam oocytes. And actually, clam oocytes were much more interesting than sea urchin eggs, because when they were fertilized, they started making new proteins. And actually, we got a Cell paper out of about 3 weeks' work that summer, just by showing that the messenger RNA for these proteins A, B, and C were already present in the unfertilized oocytes, but were not loaded onto ribosomes. And then something happened to load them onto ribosomes. So this is a really excellent example of what we now call translational control of protein synthesis. And we began, or at least I began, to wonder, why did these organisms need to make new proteins after they were fertilized? And, well, how do you know? And again, you know, you're not really thinking as clearly as you might. And of course, you need to make new proteins. You're turning from an egg into an organism. And you look at what's going on here. It's pretty amazing. These photographs, they're not taken by super resolution microscopy by any manner of means. But they're taken by the same microscopic techniques. This is before fertilization. This is about 20 minutes later after fertilization. And I think you can see there's been a huge reorganization of the cell. And there has been no new DNA, RNA, or protein synthesis to speak of. So that's all done by some kind of change in the state of the proteins that were already present in the unfertilized egg. So, you know, think about that. And so these ideas are sort of buzzing around in the back of my brain. And then that same summer, there was a wonderful seminar by this guy here, John Gerhardt, who was a brilliant enzymologist. He was really the guy who put allostery on the map, except that the French coined the term, and they got rather overlooked. Anyway, John talked about his new studies on the effects of progesterone on frog oocytes. I should perhaps explain that oocytes are the things that all the females in this audience have inside them, which will eventually become eggs. And a female frog is absolutely loaded with oocytes. And the oocytes turn into eggs as they pass down the oviduct, and they come out and they get fertilized by the sperm. And that process, which is called maturation, the process of an oocyte, a cell that's going to be an egg, turning into an egg, is triggered by the enzyme progesterone in frogs. I never sort of got reproductive physiology in humans, really. It was too complicated. Frogs is easy. And I just added some progesterone, and you see what happens. A white spot forms, and then they undergo the first meiotic division, this sort of convulsion. And what John described was that this guy, Yoshio Mizui, a Japanese worker working in Canada, had discovered was that when you add progesterone, it's a slow process. It takes several hours for the oocyte to turn into a fertilizable egg. And the fertilizable egg is actually arrested in the end of second meiotic metaphase. And this thing here is those are the chromosomes lined up on a meiotic spindle. And when the sperm comes in, then that triggers anaphase, and the chromosomes all come apart, and the second polar body is admitted. I understood meiosis for the first time in my life by watching the clam oocytes do all this stuff. And the key thing was that although the first thing was very slow, if you then sucked a little bit of cytoplasm out of that and injected it into a fresh oocyte that had never seen progesterone, this very rapidly underwent the same process. And it was pretty amazing because whatever this stuff was, and Mizui had called it maturation promoting factor, MPF, it seemed to have the ability to turn itself on, autocatalytic. So that was really interesting. But it was terribly unstable. And John was trying to purify it. And I thought, oh, what a delicious problem. If only I could study that, how wonderful that would be. But I was studying protein synthesis. So nothing happened. That was 1979. And various other facts were found about MPF. It was sort of, the pressure to find out what MPF was increased. And then I did this experiment, which was a really, really simple dopey experiment that could have been done any time in the preceding 10 years. It required the invention of one-dimensional SDS polyacrylamide gels, and you had to have some fertilizable sea urchin eggs and some S35 methionine. And I think you can easily see this protein here, and I was doing this for a completely different reason. I wanted to compare the patterns of protein synthesis in properly fertilized eggs with parthenogenetically activated eggs, virgin birth eggs. Now, this is an experiment in molecular theology, actually, to test the properties of virgin birth. And you see this protein is the first one that you can detect, and it gets stronger and stronger, and then it sort of faded away. And then it came back again, and then it faded away again. And I remembered the fact that if you inhibit protein synthesis, these eggs don't divide. So thinking about MPF and thinking about why do you need to make these, it's sort of absolutely obvious what's happening. You have to make this protein, and it catalyzes cell division. It has something to do with turning on MPF. And then you have to destroy it in order to complete the cell cycle. Well, people looked at me and I got very excited by this. And we looked in clams, and below me down, two of these proteins, and Eric and Joan hadn't discovered this. They'd been working on it for ages, but they hadn't done this really dumb, simple experiment that I did. And you can see that both of these proteins, the technique has got a bit better now. A and B are both cyclins, which I called because I was a keen cyclist. And they disappear, and they come back again. And if you inhibit protein synthesis here so they can't come back, you block cell division. So I knew I was sort of on the track of something really important. But the problem was how to find out what these things were, because we had no idea. And luckily for me, cloning and sequencing had been invented just about the same time, and it took a while to clone and sequence these things. And perhaps more to the point was that now I'd been an expert in protein synthesis, and now I was I was being led ineluctably into the control of cell division, about which I knew absolutely nothing. So what do you do if you know absolutely nothing? Well, you read and you talk to people, you show people your results, and you say, can you see anything wrong with my idea? And nobody could ever pick a hole. It was rather like Stefan Hell. It was right. I mean, it was robust because it was right, basically. But nobody believed me. I mean, everyone thought I was completely mad. And I learned about the cell cycle. And really, the cell cycle is all about distribution of DNA between cells. And there are two important phases: the time when the DNA is replicated and the time when it's distributed to the two daughter cells. And Sidney Brenner said this wonderful thing: the moment of absolute enlightenment, the reduction of biology to one dimension. It's a great thing. So, and Sidney was very keen on self-replicating automata. And so you can sort of say that what's going on here is that you have a tape which has all the instructions for making a new organism, and the trick is to put a copy of that tape in every single one of the cells in the body. So you have to copy the tape, and then you have to distribute the tape between the two daughter automata. So, there's a gap while you prepare for copying, then you do the copying, then you prepare for insertion and segregation, and that's how the cell cycle is. And there are 3 points in the cell cycle where decisions are made. And these decisions are really interesting because they're sort of irrevocable decisions. You take the decision very carefully, but once you've made that decision, you never ever go back on. You can't start DNA synthesis and then unpick what you've made and go back because you sort of ran out of nitrogen in between, right? And so so how, what were these switches like? Really, really, crunchy biological problems, and I'm sure my protein's got something to do with that, but how to find out? Well, the finding out really came, the pioneer was Lee Hartwell, who discovered that there were genes that actually controlled these decision points. And I won't go into the argument about how he knew that. And then Paul Nurse, using a different yeast, found a gene that if you knocked it out, the cells couldn't divide. But if you activated it, they actually divided a smaller than usual size. So he knew that CDC2, which was regulated, it turned out, by another gene, WEE1, which also causes cells to divide small, this controlled entry into mitosis. And I remember walking down a field in Bath at a meeting with Paul, and by this time, CDC2 had been sequenced and found to be the same as CDC28, and that was very puzzling. Why? How could the same gene control two cells, different cell cycle transitions? And when it was sequenced, it was found to encode a protein kinase. And Paul said it's not a protein kinase. We've tested it. It has absolutely no activity. And I said, but it looks like a protein. No, I don't think it's a protein kinase. It must be some enzyme that uses ATP or maybe synthesizes ATP or something like that. We were really sort of confused at the time. And then the important breakthrough really came from a student of Misui's called Fred Locher, who discovered that rather like reticulocyte lysates, if you spin frog eggs very hard, you get this beautiful pure cytoplasm. And if you add to that pure cytoplasm DNA or chromosomes, actually sperm nuclei, they know what to do with it. They assemble beautiful meiotic spindles. And actually, if you add calcium to that, which replicates what happens when the sperm arrives, they will actually do anaphase. It's really, really amazing and incredibly, incredibly beautiful. And using that as the assay, Jim and Fred managed to purify MPF, and it had two subunits, and it looked very much as though the larger of the two subunits was cyclin. And I told Jim, as soon as we have antibodies against frog cyclins, we'll send them and we'll test it. And that turned out to be what it was. And the smaller subunit was CDC2. And the answer was very, very simple, that CDC2 was a protein kinase, but it was totally inactive without its cyclin partner. And why we didn't figure that out years before, I just don't know, except that the analog, all the protein kinases who were really well known at that point, did not have activating subunits. They had inhibitory subunits. That's how cyclic AMP, it has an R subunit, which is an inhibitor. And it just never, ever crossed our minds that a protein kinase could be turned on instead of off. I mean, how dumb is that? It's, you know, it's just ridiculous, actually. So, you know, once we understood it, it was really easy. So you make cyclin, that turns CDC2 on, it phosphorylates lots of proteins. And then there's only one little problem, which is that, you know, so you phosphorylate lots of proteins and the cell enters mitosis. But now you ask the question, how many proteins? How much do they have to be phosphorylated? And also getting out of mitosis, who takes all those phosphates off? And you realize that by argument must be the case. And then you run into something that you learn in Biochemistry 101, which is that if you have a kinase which is putting on a phosphate and a phosphatase taking off the phosphatase, working at the same time, you won't go anywhere. You'll just be a sort of gray mush in between, right? If you want to fill the wash basin, you have to not only put on the tap, that's important, but you have to put the plug in as well. So if you think as a tap as the kinase, and that's all you need to understand. So this is right at the end now. I was lucky. I had probably, arguably, the best postdoc I ever had came right at the end of my career, Satoru, and we figured it out, that actually the phosphatase is regulated, and this is the experiment shows that. This is measuring kinase activity, and you see mitosis is a very abrupt thing. But you can see the kinase activity is going up. It doesn't go up abruptly. It goes up sort of slowly. And yet, you can see this is showing you when the extract actually enters mitosis. So actually, you know, it could be that it looks more like this. The phosphatase is normally on, keeping you in interphase, and then something turns it off. And then, or maybe it's that the kinase is a bit regulated and the phosphatase is also regulated. But it's clear that there must be phosphatase regulation as well as kinase regulation. And nobody had, I mean, I'd always thought that must be the case, but I'd never put anybody onto it until we stumbled on it. And to cut a long story short, what we discovered was that there was this little tiny protein here, which was a wonderful phosphatase inhibitor. And this diagram, I mean, it looks quite simple, but actually it has two very nasty features. There's a positive feedback loop here, so that when you're in interphase, the blue lines, We1 keeps CDK switched off. And as long as that's active and CDK1 is inactive, I mean, you're stuck in interphase. So it's a sort of real problem. How do you ever shift from one stable state to another stable state? And there's another one here. This phosphatase inhibits the activity that inhibits itself. So again, how do you flip from one to another? And right at the end, we've collaborated with a wonderful theoretical biologist who sort of managed to figure it out. And I don't have time to show, but you know, the analysis of this circuit, when you put in all the proper parameters, beautifully predicts how it will behave. And it's very important to have this turning off of the phosphatase as well as turning on of the kinase. And now all this is sort of, you know, in the textbook and the thing. So, but it was super fun figuring it all out. And I do recommend, I mean, the main thing in the lab is to, I think, have fun. Thanks very much.
Thank you, Sir Tim. Now we invite Prof. Ting up to moderate the Q&A session.
We're going to switch.
Oh, we're switching? I'm in the wrong chair. Okay.
Fascinating talk, Tim. Like, really, really fascinating, mind-blowing. Who's the sea urchin fans here in the room? I like sea urchin very
I'm
much.
very fond of sea urchin. Best time I ever had was in Greece, where they grow on rocks, you know.
Right.
And you go down there with a lemon in one hand and a pair of scissors in the other hand, and you just pick them off the rocks.
Yeah.
Poor things. It's sad.
Yeah, I was so fascinated about like how you started your career from haem, globins, and sea urchins, and clams, and frogs, and
It all sort of hangs together somewhere.
yeah, and I'm going to ask the first questions, but please, the audience, please come to the mic and ask, Tim as many questions as possible, since we only have him for a couple of days. Since we already have like audience there, I will save my questions for later. Yeah, why don't you go ahead and ask a question?
Hi. Thank you for the great presentation. It was super interesting. Either you're very good at presenting or you're very good at identifying very interesting problems. So I wondered, how did you identify the problems to work on? I mean, you had this presentation where you just started working on heme. Do you have supernatural intuition about what's interesting, or how does it work?
No, I just, you just hear an interesting talk and you sort of, it's more interesting than what you're already working on, so why not work on it? I mean, it took a while. I always wanted to work on MPF. Once, the minute I heard about MPF, it sounded like, I knew in my bones this was a really important problem. And it felt like an accessible one, it felt within reach. And indeed, it turned out it was within reach, but not by a direct attack, That's the thing. You have to sneak up on these things from behind and catch them unawares, right?
Yeah.
I'm, as far as, I get a lot of practice in presenting this story, so that's, okay? Right. So that helps.
Right. Was it during the undergrad, the period or PhD period that you actually suddenly got this, the great idea that, you continue to pursue on, or, yeah?
Yeah, well, we were really obsessed by this business of queuing, and we really didn't focus on the problem of how the heme worked. That came later. And actually, right. I discovered only a couple of years ago that we totally misunderstood how the heme was working. I won't go into that, but okay. Really, really interesting. Yep. I mean, I think one of the things that I discovered was that how one is bound by prejudice. Yep. People telling you things that are sort of true, but not really. Yeah. Or things that you've read and internalized and never bothered to really seriously think about. Time and time again. We were doing things which in retrospect seemed so obvious. Why the hell didn't we see them years earlier and save ourselves a lot of time and trouble and heartache? Yeah. And I don't know, it was sort of always like that. And so you just got to keep fiddling away and doing little experiments and having little side projects, and sometimes something works and it's good.
Yep. And second questions. Yep.
Yes, hi, thank you very much. I'm Will, and I'm currently in my second postdoc here in Singapore. When I did my undergrads and DPhil, I trained in the UK, and then my first postdoc was in Sweden. Now, one of the part of your talk was that you mentioned that you move back to the UK and take the pay cut, and therefore allows you to be successful. Now, that was one of the reasons I left the UK and then the EU, actually, because of the pay was so
miserable.
Yeah, to put it simply. And Singapore gave a much better opportunity in that sense. Now, my question is, I really enjoyed my time at Oxford when I was there. Should I move back to enjoy and take a almost 60-70% pay cut, or stay here and remain to my hometown in Thailand and Singapore, and therefore propel the Eastern world?
Well, maybe you should ask, how do you live on the 900 pounds per year salary?
Well, actually, it was very funny because you're supposed to pay national insurance. It's a sort of tax. Yeah. And I didn't realize I was officially self-employed, and so I was bound to pay this tax, and I had no idea that I was supposed to. So finally, the Department of Health, we call them the Department of Stealth and Total Obscurity, Health and Social Security showed up on my doorstep. Why aren't you paying this tax?
Yeah.
And I said, well, you know, I earn £800. Oh, he said, well, yeah. You do realize, however, that your pension is going to be reduced. So I get a reduced pension as a result of not paying this tax. But I mean, if I'd paid it, I would have starved, I think. But, you know, maybe, you know, not having so much money makes you kind of lean and hungry. But I think the most important thing, you know, to me, the most important, labs are very social places. And I think I regarded the lab when I was in my heyday, so to speak, as a sort of total life support system. And having good mates in the lab, people on whose shoulders you can cry, and open bottles of champagne when you make a success, are super important, you know, and a lot of firm friendships are forged in those things. So I think you need to go where you know, I always used to tell students, go where you feel comfortable. If you like towns, go to London, you know, if you like the seaside, go to San Francisco, et cetera, et cetera. But the most important thing is to work with people that you really like. Otherwise, it's miserable.
Yeah.
Really miserable.
Thank you very much. Thank you.
How do you go for a date with a £900 salary?
I didn't.
Yeah, well, I mean,
I was working pretty hard, you know, and that was the era of the Cambridge rapist. It was kind of, you know, it was dangerous to be walking in the streets late at night because the police would stop you and say, are you the rapist? You'd say no.
Yeah, I'm going to close the session. Yeah, well, yeah, I am conscious about time. We are 5 now. But I wanted to ask the last questions for myself. Like, you know, Tim, you achieved a lot in life, right? I mean, you got the Nobel Prize.
I'm very lucky. I'm very lucky.
You got the wife actually told you very wise, you know, advice around yourself if, you know, clever people. I mean, yeah, that's one of your mentors actually told you. And you've been knighted as well, and we consider this like a winners in life. And what's next for you? Like, you know, what are you working on right now?
Well, I'm totally retired. I'm 81 years old, by the way, and I don't think I have very long to live. So my job is looking after my wife and the dog. We have a new dog.
Okay. Oh.
He's a puppy, and he has very sharp teeth.
Right, I saw that. I saw that. Right.
It makes dressing, putting on the socks in the morning, absolute hell. It's such a luxury to be here because there is no dog biting.
Yeah. Okay, great.
Okay.
I mean,
very good.
Great sessions, and thank you once again for coming to Singapore.